Thank you very much. I would like to start by acknowledging my opportunity to be here and to talk about this topic that is probably quite different from the others that we have been hearing this morning, but that is also something that is of concern. I will talk about a form of emerging contamination in water that is antibiotic resistance. Is something that probably it was not discussed several years ago, and that nowadays is we are accumulating evidences that that indeed antibiotic resistance is a problem in the environment. Why is antibiotic resistance a problem? When we talk about antibiotic resistance, and normally when we hear, even in the communication to discuss this problem, oh, sorry, now it's working too much. Normally, we always refer to the hospital and to patients that are in the hospitals. And we know nowadays that, and this is taken from the report from 2014 from the World Health Organization, about the real threat that is the problem of antibiotic resistance. And we realize that it is no longer a problem that is confined to hospital settings or to patients. It is also already spread in the environment. And before talking about water and antibiotic resistance, I would like to say a few words about the nature of antibiotic resistance. It emerges and it evolves very, very rapidly. Here we can see the dates when some antibiotics were available in the market, and below we can see the short period of time that was needed for resistance to become a problem in the clinical settings. Antibiotic resistance can evolve very, very rapidly, and this is really frightening. Why does it happen? We have two different processes. One is that bacteria can transfer antibiotic resistance among them. So when they are under what we say a selective pressure, when bacteria are endangered by antibiotics, they can share the resistance genes to other bacteria in the community. And this can happen in our gut, but it can happen also in the environment. And another process that is very, very important is the selection. When bacteria are under the pressure of antibiotics, only those that are resistant will survive and the others will die. So these are the major mechanisms that happen always that we have antibiotics and resistant bacteria together. And for this reason, antibiotic-resistant bacteria and antibiotic resistance genes are nowadays considered, or can be nowadays considered, contaminants of emerging concern. But they are a special type of contaminants. Why? Because they can auto-replicate in the environment, they can auto-proliferate. So they can spread long distances, for instance, through wildlife and through water, and they can proliferate. So they are a very special form of pollution. Is biological pollution, and biological pollution can auto-replicate, and it's really a problem. The urban water cycle has everything to do with the spread of antibiotic resistance. In this cycle, we have two major parts. One that is the, let's call it the unclean part, where we have the water that results from our use, from our consumption. This water is normally collected, is treated, and then is again delivered to the environment. And when we do all this process, we hope that the water is clean, is good enough to go again to the environment. And probably is not always like that. On the other part of the water cycle, we have the water that is abstracted from clean water sources is normally disinfected and is distributed for us to use. And in both parts of this cycle, we have evidences that contamination with antibiotic resistance can occur. Let's say a few words about wastewater treatment plants. Here we have the ideal conditions for antibiotic-resistant bacteria to spread and to proliferate. Why? Because here are discharged very high doses of antibiotic-resistant bacteria from humans, from animals, which come from our houses, from hospitals, and from industry, even if they are pre-treated before they are discharged in the municipal collector. Here we have also antibiotic residues, and we have several other contaminants. And this mixture is probably very, very good for bacteria to survive, mainly for antibiotic resistance to survive. What we can see from different studies that were conducted with different wastewater treatment processes, what we can see is that treated water that is discharged in the environment, treated final water, it will contain 10 to 9 antibiotic-resistant bacteria that is discharged per minute. This is a huge amount of antibiotic-resistant bacteria that is being discharged continuously, 10 to 9 per minute. And this is an underestimation of the real number, probably. So this is a reality, and this is a reality that apparently is occurring everywhere. Sometimes it happens also something that is not good. Some types of antibiotic resistance can even increase during the wastewater treatment process. Why? When I say increase, I'm saying that the proportion of a resistant bacteria will increase, will be selected during this process. And this is also something that can be really a problem, because instead of cleaning, we are promoting resistance to spread. When we compare, and here in these studies, we used Escherichia coli, that is a fecal contaminant. We compared, based on multi-locus sequence typing, we compared Escherichia coli from different sources, from urban wastewater, from hospital effluent, from patients, from urban streams, wildlife that live in our city, for instance, in Porto, where I come from, and from wildlife that live in a natural reserve. And what we could see is that these bacteria that share these different environments, they are genetically closely related, which means that probably this contamination that comes from hospitals, it is indeed already found in the environment. These birds that are, that are found, that live in a, that live in a natural reserve, they have a different lineage of antibiotic-resistant of bacteria, of Escherichia coli, which means that indeed it's the human impact that is originating this contamination. Okay, if we see that these bacteria are spread everywhere, a logical question that we can make is, okay, are they present in drinking water? Can they reach our tap water? In principle, we should think, okay, the water is disinfected, so it will be free of antibiotic-resistant bacteria. But what we can see is that when water is disinfected, we have a real important reduction on the number of bacteria in water. But in reality, when we analyze the tap water in our houses, we find that indeed we have a lot of antibiotic-resistant bacteria, although this bacteria has something that we call intrinsic resistance. It's something that is natural in bacteria and is not acquired by those processes that I mentioned. However, when we analyze the total DNA, which means that we are not cultivating bacteria, we are directly analyzing the DNA extracted from tap water, we find some acquired antibiotic resistance genes. So it means that probably it is a very residual contamination, but it is already there. We don't know, however, the consequences of this contamination of drinking water for human health. Obviously, we don't know if these genes are indeed transferred for us or not. It's a question that we still have. Okay, when we look to these results, we think, okay, it's time to start doing some things. By one side, we need to control if resistance can go from the environment to humans. This is a major question that we have now. We don't know if it occurs, how extent it is, we don't know. And on the other side, it is very important to control this form of contamination, of environmental contamination. So we started working on that. And now I would like to share with you some networking activities that we have been doing. In a cost action that we have that finished in 2013, we were more than 100 scientists from different areas, and our topic was antibiotic resistance in the environment. And we discussed largely what could be done, what is missing now to control this problem. And we went up with a position paper which suggests that there are some things that we can do now, and we must do it now. One of the problems and one of the gaps that we have nowadays is that we have a lot of data, but no data is comparable. So we need urgently to harmonize data and to share this data among countries. This is important for us to know if there are different regions in the world with different patterns of resistance, how this resistance is related with the chemical contamination, and also if we implement some measures, if these measures are efficient or not. So we need to have some standardized methods. Another thing that we need to do is to know if there is a risk of transmission of resistance from the environment to humans, and if this risk exists, how can it be controlled? And for that, once again, we need data. And then we need also to define some points where we can control the spread of antibiotic resistance. Hospital probably is one important source. Animal production is another one. So we have some critical points where probably it would be very, very important to improve the treatment processes. For instance, by implementing advanced treatment processes. In order to go ahead with these initiatives, we have nowadays some activities. One of these is a project that we have already started last year. We are trying to initiate a comparable set of data on antibiotic resistance occurrence in treated effluents. This is made under the scope of Working Group 5 of the Norman. Norman is a network of research and reference laboratories. And we analyzed, we are different countries, I think 20 or something, from the west to east Europe. The eastern country is Israel. And when we compare the data, and I'm not going into details now, but we realized that some resistance genes that are known environmental contaminants, they are indeed in every wastewater treatment final water that we analyze. So the question now is not where are the antibiotic resistance genes, they are everywhere, and how can we control them? Another, this is a Water GPI project under the scope of Water GPI, STARE, that stands for Stopping Antibiotic Resistance Evolution, is also related, and we hope that we will have a very nice overview of antibiotic resistance in Europe. We hope we can launch for the first time a database on antibiotic resistance genes and antibiotic residues occurrence in drinking water, in wastewater. Another cost action, this one in wastewater reuse. You have the site over there. It's a huge cost action with different working groups. Everybody is welcome to visit this site and to join us. And last but not least, a PhD training school that will happen in it will start in October, and again, if you are interested, there will be 15 PhD positions available in this international training school. So you can apply. Without money is not possible to do work. We have some of the partners in these projects. And that's all, and I'm sorry if I took too long.
Thank you very much. Obviously, we have lots of activities in this area. Just one very quick question and quick answer. Just one, yes, sir. And while you're talking, can I ask for the next speaker to be ready during that time?
Okay. Question, yeah. Is there a regulation or guidelines for antibiotics in water?
Sorry?
Guidelines for antibiotics in waters?
No. One of the reasons we are working on this database is to gather some data, is then to be able to draw some guidelines.
So to can judge that it's polluted or not polluted, yeah?
Yes.
Yeah.
Yeah. And also, if there is a risk of transmission from water to humans, that is important for this assessment. Yes, of course.
Okay, thank you.
Thank you. Okay, we continue with the next speaker.